All right, let's get right into it. We usually think of evolution as this super slow, almost glacial process, right? Something that takes centuries. But in the world of bacteria, things are happening at an absolutely insane speed. It turns out they have this kind of secret social network for sharing genes, and figuring it out is one of the biggest challenges in medicine today.
So that brings us to the big question. How does a bacterium that's basically harmless suddenly become a deadly antibiotic-resistant superbug? It's not just some random 1 in a billion chance. Nope. There's a really powerful system at play that lets them adapt, well, almost overnight. And that's the mystery we're going to unravel right now.
Okay, so the evolution we all learned about in high school is based on mutation. It's slow, it's gradual, it's totally random, and a lot of the time, the changes are actually bad for the bacterium. But bacteria have a shortcut, a genetic cheat code. Instead of just waiting around for a lucky mutation, they can grab huge chunks of proven, battle-tested DNA all at once. This gives them an immediate leg up, like instant resistance to a drug. Think of it like this. Mutation is trying to invent a hammer from scratch, while the shortcut is like getting a fully stocked professional toolbox delivered to your front door. And this incredible process, this great gene swap, has a name. It's called horizontal gene transfer, and believe me, it is a total game changer. Now, here's what makes this so wild. This is not your standard inheritance, from parent to child. Horizontal gene transfer, or HGT, is more like passing a note in class. It's a direct swap of information between two individuals, sometimes even between totally different species. It basically rewrites the rulebook of evolution. And just how big of a deal is this? Well, get this. Scientists estimate that a full 20% of the entire genome of E. coli, one of the most studied organisms on Earth, didn't come from its direct ancestors. It was picked up from other bacteria through HGT. I mean, just let that sink in. 1/5 of what makes an E. coli was basically crowdsourced. It really shows you that for bacteria, survival is a team sport. So you're probably wondering, how exactly do they pull this off? Well, bacteria have a whole bag of tricks, not just one method. There are 3 main ways they swap genes: transformation, transduction, and conjugation. Let's break down each one. First on the list is transformation, and you can pretty much think of this as genetic scavenging. It's a little morbid, actually, but hey, it works. So, it all starts when a bacterium dies. Its cell wall breaks apart, and its DNA, its entire genetic blueprint, just spills out into the environment in little pieces. This basically creates a public library of free-floating genetic code for any other bacteria in the area to browse. So next, a nearby living bacterium can enter this special state they call competence. It's like it flips a switch that says, okay, I'm ready to absorb some DNA. And if it finds a useful-looking piece floating by, say one that has the gene for antibiotic resistance, it can literally just pull it right inside itself. And for the final step, that scavenged piece of DNA doesn't just hang out. The bacterium actually weaves it directly into its own chromosome, making it a permanent part of its genetic code. And just like that, poof, it has a brand new trait. It is officially transformed. All right, on to the second method, transduction. This one's really interesting because it involves a middleman, a virus that acts like an accidental delivery driver for bacterial genes. And that's the key thing to remember here. The virus doesn't mean to do this. The whole thing happens because of a tiny, random screw-up when the virus is trying to make copies of itself. So here's how it plays out. First, a type of virus called a bacteriophage attacks a bacterium. It takes over the cell and starts making more viruses, and in the process, it chops up the host's DNA. Now here comes the mistake. As the new viruses are being put together, one of them accidentally gets stuffed with a piece of the bacterial DNA instead of the viral DNA. Then this faulty virus goes off and infects a new bacterium, but instead of injecting a virus, it injects a piece of DNA from its last victim. And you can see exactly what's happening right here. On the left, you've got your standard issue virus carrying its own viral genes. But on the right, that's the transducing particle. It looks the same from the outside, but its cargo is completely different. It's basically a Trojan horse, but it's filled with bacterial genes instead of soldiers. And that brings us to our third and honestly our most important mechanism, conjugation. Now, this isn't scavenging, and it's not a viral mistake. This is a direct, deliberate transfer of DNA between 2 living bacteria. And I really can't overstate this. Conjugation is the superhighway of the bacterial information network. It's the #1 way that powerful traits, like antibiotic resistance, spread so darn fast, even between completely unrelated species of bacteria.
So how does this direct connection work? Well, one bacterium, the donor, grows this long hollow tube called a pilus. This pilus then reaches out and attaches to a recipient cell, forming a physical bridge between them. It's a direct pipeline built for one thing: shooting a copy of its DNA across.
The genetic machinery that makes this possible is just brilliant. The instructions are on a special little loop of DNA called a conjugative plasmid. Think of it as a USB drive that has a program on it to force the next computer to copy it. It's got trad genes, that's the toolkit for building the bridge, and an ORI-T sequence, which is basically a big sign that says, "Start copying here."
Okay, so we've looked at the how. We've seen the different ways bacteria share their genes. But why does this microbial social network matter so much to us? Well, the answer is pretty straightforward. And honestly, it's terrifying. This is how superbugs are made. And this right here really gets to the heart of the problem. The very plasmids getting shared through conjugation are often something called R plasmid. The R stands for resistance. These things carry the genes to defeat not just one, but multiple antibiotics. A single transfer can turn a totally treatable infection into a life-threatening, multi-drug resistant nightmare.
And look, this isn't some rare, abstract problem. This is a massive medical crisis that affects how we treat infections from really common bacteria. We're talking E. coli, Salmonella, Pseudomonas. The names you see on this list represent some of the biggest challenges doctors face in hospitals every single day.
So, if you take one thing away from this, let it be this: horizontal gene transfer is the engine that's driving the superbug crisis. It allows a single resistance gene to just sweep through a whole population of bacteria with incredible speed, pretty much like a wildfire tearing through a dry forest.
You know, it really forces you to look at bacteria differently. They aren't just these isolated little germs. They're a huge, interconnected community that is constantly sharing information to survive. They basically invented the social network billions of years before we ever did. And that kind of leaves us with one final thought. With their incredible, relentless ability to adapt and share information, what can they teach us about our own survival?